everybody. Welcome back to Psychedelics Today. This is Joe Moore coming at you from Breckenridge, Colorado. Today on the show, we have Paul Daley. Paul Daley was working with Sasha Shulgin for years in his lab. Paul is a very interesting guy, and we got to record, thanks to the Shulgin Foundation, at the Shulgin farm in Sasha's office, which was amazing for me. And in some cases, made for some really great videos. So check out what we've done in partnership with our friends over at Hover. Really epic videos on our YouTube, honestly. So please do check those out. It's super cool. And yeah, Paul is also heading up the Shulgin Research Institute, where they are actually doing the Shulgin process, developing new molecules and trying to bring them across through a normal FDA route. So slightly different from what we would see Sasha doing, which is testing on himself and testing with some others, reporting the results. But I think it's important. There's a lot of molecules that definitely do need to make it into the pharmacopoeia. And I think the name of the group is the Alexander Shulgin Research Institute, ASRI, Psychedelic Discovery Development. And it's great. So thank you to our friends at Hover. Ethan was there making some epic video for us. And then the Shulgin Foundation for making these interview slots available for us. I really appreciated it. And thanks to Paul for sitting down with me as well. Really lovely. So I think that's it for now. So thank you all for tuning in. And thanks again to the Foundation and Hover for making this one happen. And we'll see you on the other side. Enjoy. All right, here we are, Psychedelics Today, joined by Paul Daley. How are you today?
I'm doing fine, thank you.
Great. So we're in Sasha's old office, and
right.
Beautiful day here.
Right.
So how do you fit into this whole Shulgin puzzle?
Well, I'm kind of the certifiable graybeard. I met Sasha at a meeting in 1977.
Mm-hmm.
I was a freshly minted Ph.D. student at UC Berkeley in entomology, of all things. And when we met, this was at the second International Conference on Hallucinogenic Mushrooms, an event that I was kind of unaware at that time. That,
at Berkeley?
No, it was in Washington State,
okay.
Port Townsend. I had arrived at Berkeley in July, I think, and ran into an ad for this meeting. And, I had, was just going through kind of a life upheaval, getting into Berkeley. And I was thinking, well, how would I explain going to this to my faculty advisors? And so at first I thought, I couldn't do it, but then I saw the ad again a couple of weeks later, and, Albert Hoffman, Gordon Wasson, Richard Everett Shulgin. I thought, I don't know that these gentlemen are even going to be alive very much longer. So I bought my ticket. And the second person that I met at the meeting was Sasha. And
so this presumes you had some experience in the Bay and you were kind of aware of what was going on in the psychedelic world.
Actually, I had a first little taste of research in psychedelic tryptamines 5 years earlier. As an undergraduate, I orchestrated an internship to work on tryptamines in phalaris grass. So, totally from the perspective of them being like a toxic problem for animal husbandry, and but my motivation, of course, was to learn how to handle psychedelics analytically. So it was very early in my college career, and it kind of went away because the Nixon war on drugs was just being lifted off the ground at that same point, 1972. So when I met Shulgin, he said, well, so why are you here? And I explained that I was fascinated with psychedelics. I was interested in analytical toxicology, and had studied phalaris. And well, maybe you can tell me about this. And so we just had a lovely animated conversation. And he's a very impressive character. He had an air about him that was quite fascinating. And of course, the third person that I met in that little group at the wine social turned to me and said, "Oh, you're interested in analytical chemistry. You might want to come and visit my lab. I'm the director of the Drug Enforcement Administration Western Analytical Lab in San Francisco." So I kind of went gulp. And the thought that went through my mind was, I've either arrived or I'm really in trouble now. So, but turns out that was Bob Sager, who had already become quite close with Sasha, and his story has already been documented. So, anyway, but Shulgin being a Berkeley grad, and he said, "Well, you're practically in my backyard, so come for a visit." So probably around November of 1977, I walked down the path to the lab for the first time. And, it has a real charm to it that's quite unique. So, that it stirred my imagination. Our lives diverged from there. I was busy wrapping up my thesis work. I did a postdoc in Quebec City for a couple of years, so that took me out of the area completely. So I missed his wedding in '81. Came back to California with a postdoc out at the Lawrence Livermore Lab, which is a large Department of Energy, national research lab. And I was working on vegetation physiology, responsive vegetation to elevated atmospheric carbon dioxide. So, yeah. So I was a plant scientist, and we were kind of one of the first groups exposing plants that were long-lived, perennial species, to elevated CO2, and the results were very interesting and kind of controversial. But after that program kind of wound down a couple of years later, I was able to get hired on as a staff scientist and worked basically in environmental restoration projects for the next 20 odd years. I was out there a total of 30 years and did a lot of instrumentation development, kind of field engineering of cleanup systems, some outside work for the Navy, for the Army. But psychedelics always kind of pulled me. So in the early 2000s, I started visiting Sasha again, and kind of at that point, a scene had sort of emerged. He had a lot more visitors. First couple of times we met, it was really just the two of us out here, and the second time, in fact, was kind of there was a tragedy in our department. We had a very charismatic, fun director of our program in entomology, who was a specialist in classical biological control, so basically ecological pest management. And he had published a book, The Pesticide Conspiracy. This is Robert Van Bosch. And he was really, taking a full slam at the chemical industry and how they were really wrecking agricultural ecology. And in the week of the Jonestown massacre, that news came to the Bay Area on the Sunday. This would be the third week of November in '78. And since there were so many people from the Bay Area that were down there in Guyana, that really hit very hard. Well, anyway, Van Bosch was out running on the Sunday, fell down dead. So we came to work on Monday morning, and that was the news. Apart from Jonestown, Van has died. And at the end of that week, of course, was the George Moscone Harvey Milk murder. So it was a very dark time. And the department, there was I think an honest concern that there might have been foul play in Van Bosch's death, particularly because he had just had a clean bill of health a couple of weeks before he passed. So knowing that Shulgin taught forensic toxicology, I phoned him up, explained the situation. He said, well, if unless he was very elderly or in the immediate care of a physician, there would automatically be an autopsy. If you really think there's a possibility of there having been foul play, that's a public record. Get a copy of it, bring it out, and we'll go through it. So the second time I had an afternoon with Shulgin here at the farm, we spent the afternoon going through an autopsy report. And ultimately there was silent heart disease that in that era was not picked up, so there was really nothing to hang our hats on. Of course, by the end of the conversation, Sasha would always kind of, with a twinkle in his eye, kind of take things in an entertaining but dark direction. He said, "Well, if you wanted to kill someone chemically and leave no trace, how would you do it?" And I won't go into those details.
Right. Thank you.
Yeah. So anyway, in the sort of early 2000s, we kind of re-upped our relationship, and like I say, I started visiting, not to do chemistry. I thought it would be too presumptuous to offer to help. But by 2007, Sasha actually performed the wedding ceremony for my wife and I here at the farm. And I happened in on a conversation during the ensuing party between Anne and her buddy Tanya Manning that they were having regarding a benefactor who had stepped forward and offered to fund a half-time position for a chemist to work with Sasha and keep the work going. So long story short, I was able to work out a deal to drop back to half-time at Livermore. And so half of the week I was cleaning up after the bomb boys, and half of the week I was working with Sasha. So for the last 7 years of his life, I helped kind of put the lab back in working order, helped him with kind of a pet writing project that he had going for about 10 years, the Shulgin Index, which was a large reference work on psychedelic phenethylamines and amphetamines, which was his primary original research contribution. And just kind of helping out, and it was a delightful time in a lot of ways. It was very challenging. His decline in health was very distressing, but we all tried to do what we could to make his life comfortable and interesting. And so I subjected him to a lot of strange music out in the lab and conversation about how weird politics had gotten, and we had a good time.
It's amazing. And now you're in a way continuing the work with the Institute.
Right.
Can you talk about that?
Sure. So a partner of mine, Nick Cozy, who's a retired pharmacologist from University of Wisconsin, who actually Sasha introduced to me in 2003, we had been publishing kind of off and on in the ensuing years. And in 2019, we published a paper on production of high purity DMT, hemifumarate, for clinical studies, with kind of a full pharma style characterization of the substance, its synthesis, its purification, characterization. And right around that time, the sort of investment interest and public interest in psychedelics was starting to really rev up, and we started getting inquiries. And make a long story short, we were able to get some seed monies from a Canadian venture capital firm, and that enabled us to actually lease a lab, incorporate the Alexander Shulgin Research Institute, and kind of get it off the ground. So we're in our second year. We have at least one lead compound that's one study away from being ready to go into clinical trial, which we're pretty excited about. And we're kind of advancing what Sasha laid out as a foundation. So Sasha was really primarily a drug discovery scientist. So his interest was in discovering new structures and doing upfront characterization of threshold doses required to produce any effects at all, and then exploring, what doors were open by his different inventions. But development from a pharmaceutical point of view was not really something that he was interested in. So we've actually pushed more in that direction. So we're taking the time and the marshaling of resources to move compounds through more molecular pharmacology characterization, animal toxicology studies, and really getting them ready to go into formal clinical trials that would be FDA approvable. So that's where we are now.
And mostly around psychedelic and psychedelic-ish compounds.
Right. Mm-hmm. Well, we've tried to, knowing the base of compounds that he developed, we have also looked at the landscape as it stands right now, and really there, in any drug development, new push, there are going to be aspects of the available drugs that could be approved upon. So duration, manageability in terms of intensity of the experience, all the things that are challenging for the psychiatric community are things that we can at least attempt to address by design of new compounds. So our most advanced compound right now is a tryptamine that's orally active, fast onset. The latent period before you start to feel effects is as little as 6 to 8 minutes. Relatively short acting, 90 minutes to 2.5 hours, and satisfyingly internally psychedelic, as opposed to like entactogenic. We're all familiar with the MDMA class of effects, but interestingly, no visual involvement. So there's no open-eyed, closed-eyed visuals. That aspect is just not present.
That's super interesting.
Yeah. And we think that there's probably a subset of patients who are, for whatever reason, reluctant to get into a full immersion psychedelic experience, at least initially. So, once they're more familiar with the territory, something like a psilocybin experience, or, something more intense and all-encompassing might be more easily tolerated if there are some training wheels in advance.
Right.
So we think that compound has that role. We have a second lead compound that's a phenethylamine that is actually not psychedelic, but it does have effects on mood, concentration, focus. And I've been saying that I think it sort of captures the effect that people are trying to achieve with microdosing, but on two accounts, it has a much better safety profile. If you push dose, since it has limited efficacy, that's how fully can the receptor systems that it touches be turned on. Its efficacy appears to be low, so even if you push dose, there's sort of a ceiling of effect, and it never becomes really psychedelic. So that's a safety factor. You can't overdo it and be all of a sudden tripping at your desk, which is not necessarily a great outcome. And the second factor is, I don't know if you're familiar with the serotonin receptor subtype 2B. I think we'll be hearing more about that in that it's sort of an off target, if you will. It's a receptor that's highly expressed on heart valves. And if you...
This is like the fen issue.
Exactly.
Ph.D.
Exactly.
Like that,
yeah. Methosergide, lysergamide, migraine drug was taken off the market for the same effect. If you're constantly turning that drug on with, or turning that receptor on with a drug that's administered quite frequently, daily or multiple times a week, you run the risk of having the heart valve thicken and become ineffective. So it's a reversible condition, but nonetheless, it's serious. So unfortunately, LSD and psilocybin turn on the 2B about as effectively as they turn on 2A, the one that seems to be required for psychedelic effects. So not really a problem if you're talking about intermittent exposures, but if you're talking about an everyday or every other day type of drug, it's a risk factor.
Mm-hmm.
So this second compound that we call ASR 2001 is about 100-fold less effective at 2B than at 2A. So, now, is this useful as an ADD drug, or is this for the betterment of life on planet Earth? That remains to be seen. The FDA wants to assure that new drugs are safe and they have efficacy against some disease. So, in the case of how microdosing is being deployed, are you treating a disease, or are you just trying to improve life?
Mm-hmm.
Improving life is not necessarily the FDA mandate. So, that'll be a challenging one. But
that's interesting.
Yeah.
I've thought a lot about this heart valve situation. I'm sure you have as well.
I think we're going to be hearing more about it.
So my intellectual shortcut, because you have to come up with something eventually, was, wouldn't we have seen it in the Grateful Dead scene a lot more, or are we?
Well, the Grateful Dead were never microdosers.
Right. They're like extraordinary macro people.
Right.
Even Amanda Fielding, for instance,
Sure.
Pretty high doses daily.
Yeah, that's a great question. So the studies that have been done to date that actually have exposed this heart valve situation have been with MDMA. And there's one study out of Europe that was pretty large. Some thousands of people were examined, and with echocardiography, you can detect the dysfunction of the heart valves. And they found now, of course, this is with people who are taking MDMA like at least once a week and sometimes more, and at larger doses. But the rate of pathology was about 28%.
Whoa!
So that's the best evidence that we have now that this kind of thing can occur. So we just need to learn more about it.
We need a lot more science, no doubt.
Absolutely.
Yeah. So I'm glad you're here doing it. And it's like, we will need alternatives, right? Like, obviously we want safer drugs when we can have them.
Right.
Always. And just reduces the harm profile and much more.
Right. And of course, to get at those kinds of questions, we now have the tools to do that weren't available to Shulgin when he was doing the bulk of his work. Cloned cell types that express receptors specifically, but it costs to do those studies. So of course the financial situation is quite different from what Sasha was faced with. So
right, his interest and project was so different.
Yeah, absolutely.
Yeah, and that's fascinating. Have you heard of this guy, Dr. Z, Ezekiel, out of Vancouver?
The only Dr. Z I know is out of Israel, so I probably don't know.
He moved.
The same person? Okay, yeah,
Pre-MMC, for MMC.
Yep. Okay, we have met.
Yeah, wild guy. He claims to be a neo-Shulginist. It's a very different project.
Sounds like the Dr. Z that I met. Yeah, very good.
Yeah, he's still active, doing weird stuff up there. But it's fascinating to know that there's like multiple projects. He's certainly not taking the biotech approach, as far as I understand. Yeah.
No, you don't. You don't have to. That's probably not a likely route for that bunch of compounds.
Right, for sure. So I was unaware of MDMA hitting that receptor site and having a VHD complication, and that's a really fascinating issue, given like where we're at with the MAPS FDA situation. And
yeah, and of course with MDMA, since it's probably not hitting the receptor itself, but just flooding the body with the endogenous stores of serotonin. It was serotonin indirectly is doing the work. It's like that's the substrate for that receptor. So I don't have the numbers off the top of my head, but I would imagine that MDMA probably doesn't bind to the 2B receptor itself, but it just indirectly promotes a lot of loose serotonin floating around that in fact would hit that receptor and turn it on.
Well, that's interesting. So a lot of like these more popular substances could actually be impacting that in some way or another.
Absolutely.
Wow. Okay.
So, but fortunately, like I just this morning read a paper describing software and cell systems that were being used to evaluate anti-inflammatory effects on heart rhythm. And so there's now a fairly well-developed line of being able to create cells in a petri dish that represent heart tissues, and these cell lines can be very specific to different parts of the heart. And in fact, they were talking about cells that are pacemaker cells, so the cells that are involved with controlling rhythm. And these can now be probed in a dish, and so you can screen drugs for that might have some other beneficial target, but you want to avoid having a heart arrhythmia side effect that may only show up after you've started dosing many patients. So it'd be nice to be able to avoid that.
Right.
And we're making a lot of progress in that direction.
Great. And that's good for biotech largely too.
Sure.
Not
absolutely.
Any drug could come to market and get canned 6 months, 9 months later, which would be horrible.
Right. Well, I didn't realize that the number of fatalities from side effects of NSAID anti-inflammatories was as high as it was, but apparently some tens of thousands of people have lost their lives to what we think of as being benign drugs. So there you go.
And so thinking like ibuprofen, right?
Exactly.
Yeah, that's interesting. Yeah, right. And we do want to take those risks into account. So I guess since you were able to spend so much time with Sasha, presumably Anne as well, like how do you see kind of their legacy playing out over the next handful of years here, maybe with this foundation, with the farm, or maybe in what you're doing?
Sure. Well, yeah, the foundation and the ASRI are independent, but of course we're linked because we sprang from the same wellspring. I think the challenge going forward is really going to revolve around communication and education. My fondest desire would be to see a situation where access to psychedelics became liberalized to a great degree, but with the understanding that education about how to use them beneficially, how to avoid risks. Any drug that has as profound effects as psychedelics will have some risks. But if you educate yourself in terms of the nature, the dose, and I hate to say it in a way, but there probably will have to be some regulation. That'll be a way that we can proliferate the access and improve the overall safety. So those are the challenges. Hopefully, we can keep our wits about ourselves and figure out a way to kind of get those messages out. I kind of kiddingly say that we need for people who are completely naive, a driver's education course where there's theory, and then eventually you get behind the wheel, and eventually you get your permit, and so that might be a little much to ever see in reality, but if it can be started informally and then pushed forward as sort of a regular part of coming of age, I think there a lot of difficult situations can be avoided, and the more positive aspects can be enhanced. I always recommend Aldous Huxley's Island. I think that's a model that we can aspire to. Really having psychedelics be a useful rite of passage that's accessible.
Here's a niche question about with Sasha. It seems like he was a fan of tasting the chemicals, to like really know the soul of it. I think that's how it was put. Do you have any perspectives on that?
Well, yeah, recall that when he got started, there was no molecular pharmacology really. The only way to really assess a psychoactive drug's effects was self-administration. And there's a long history of self-experimentation as a legitimate route for discovering new drug action. And there was no, also when he started, there was no stigma about psychedelics per se. They were viewed in the chemistry community as being potentially amazingly valuable and novel substances for use in treating a whole variety of mental illness situations. So Sasha, however, did, I think, pay a price. I think he's, in fact, talking about the heart valve situation, he's sort of the poster child for a bad outcome. In his 80s, he had to have a heart valve replaced. And that was, we were warned ahead of time that cognitive decline is a not uncommon effect, particularly in elderly that have procedures like that. And we did see that. So he was, of course, he was unaware, science in general was unaware of the potential risk from psychedelics. They were pretty much viewed as having a good safety track record, but that was based on natural products like mescaline and psilocybin. So as you get into synthetics, small differences in molecules can have large impact on both their intended use and off-target effects.
Yeah. Well, thanks for sharing that. That makes a lot of sense.
Yeah. It was hard to see, and so his quality of life was affected, in his later years.
Right, and we certainly want to avoid that kind of suffering when we can.
Yeah, I think we're, as our understanding improves, we can reap the benefits and avoid the pitfalls.
So super briefly, do you, as somebody involved in biotech, have any hopes for how AI or large language models might be applied to drug development, safety, and more?
I every day have like a dozen emails that are talking about the latest advancement in drug development, drug identification via some combination of sort of physical modeling of targets, modeling of receptor structure, and yes, I think there's huge promise. But so far as ASRI is concerned, we've talked to a couple of companies, and we haven't really tried to partner with any yet. We're still looking for the people that have the right kind of mix of experience and yet interest in psychedelics to be able to take the kind of database work that we're developing right now that's strictly kind of based on the chemistry side. And advance it into this area. Yeah, it's kind of wild.
Amazing. Well, Paul, thank you so much. Where can people find more out about your organization?
We have a website, www.shulginresearch.net, and we're lab monkeys, so we're basically pretty easily accessible.
Amazing. All right. Okay. Thank you so much. Appreciate your time.
I enjoyed it.
Great.
Okay. Take care.
Thanks. And there you have it, Paul Daley. I hope you loved that one. I thought we got into some really fun concepts and some good history as well. So if you liked it, please leave a review and share with a friend over at Apple Podcasts or Spotify or whatever you use to listen. We'd love a review. And we'd also love it if you share. And thanks again to Hover and Paul Daley and for the Shulgin Foundation for helping make this stuff happen. So thank you all, and we will see you on the next episode. This is Joe Moore signing off. Bye-bye.